Great. All right, welcome everyone to the Laney College Biomanufacturing Program. We are having a wonderful guest speaker today who's been a part of our program now for, going on 2 years, I think, Ron Shigeta. I will spend just a little bit of time introducing him. He is co-founder of Indie Bio, which is a startup accelerator in San Francisco. I think they're having their demo day today and tomorrow, in fact. You could probably find online. And he also founded a company called Wild Earth, which some of you may have heard about or seen on Shark Tank. And now I've kind of lost track. Ron is involved in so many startups at one time, and he'll probably talk about some of them today that I can't really keep up. And we probably talk every couple of weeks at least, and he's doing so many things. So I wanted to welcome him today and also let you all know that we're starting a synthetic biology certificate in the fall of 2021. And what Ron's talking about today is synthetic biology, the heart of what's driving the industry in the San Francisco Bay Area and around the country and world is all the techniques that Ron is going to cover and more. So I hope you're all excited to hear from Ron. He's going to do get into some of the tools today. So I hope you had a chance to find the link for SnapGene. If you check out the chat, I have a link for a free SnapGene download. It's the licensed version. And then also in the chat, there's a link for some plasmid maps that Ron will be referring to today. So if you want to follow along and do some of the molecular biology, please check out those links. And maybe while you're listening, download those different files and sources. Okay. So with that, I'll welcome Ron. Go ahead and take the driver's seat, Ron.
Thank you, Doug. All right, so we're going to start with the slides. All right, we're going to talk about producing protein from microorganisms. It's a somewhat different field than you guys have been talking about. It doesn't involve animal cells or mammalian cells at all, and that's because they're a lot easier to handle. Those microorganisms are a lot easier to handle. I'm going to start from the ground zero and build it all the way up. So I hope everybody recognizes central dogma, but basically, if you understand the central dogma very well, this is the most important thing about molecular biology. If you understand this really well, it'll guide you through most of what things are happening with synthetic biology. We all have DNA in our chromosomes. Every organism in the world that's alive has a DNA chromosome, and all of them are transcribed. That means copied in writing from DNA to RNA. And then the RNA is translated, that is like translating different languages, into protein by a complex called the ribosome. Okay, and when it does so, the ribosome reads 3 bases at a time and just like processively goes to the next triad and creates and puts out 1 amino acid on the protein chain. Okay. I am open to questions at any time. Is everybody following this so far? I see there's 4 people. I can see them shake their heads yes. So I'm assuming all the quiet people also are shaking their heads yes. But please stop me at any time. So RNA and DNA, they differ only very slightly in their chemistry. There is 1 oxygen that is missing in RNA that is present in DNA. Because that oxygen is there, DNA tends to form these double helices where there are 2 strands, one going one direction, the other direction going the other direction, and they form reverse complements with, as you can see in the lower right-hand corner, the thymine, adenine, cytosine, and guanine pairs pair up. And so the complementary sequences form double strands. The double strands are heavily protected, and it's a very robust polymer that doesn't really break very easily. RNA does not, because it's extra flexible, it doesn't really form long double-stranded pairs. It forms some structure, like you can see in these hairpin loops. So it has some 3-dimensional structure, but it's not predictable, and it's easily broken up by enzymes to get at it. RNA is simply doesn't have reverse complement to itself typically, and it just sort of acts as a long chain. And they're transient. They don't live very long in the body in the cell before they're broken up and recycled. Okay, so I know Doug uses this as well. Can you guys see the video? Okay, good. So this is the I don't know if you guys can hear me. I'm just going to turn off the sound. So the DNA transcription, copying off the genome, starts this way, where there's a bunch of proteins bound to a specific sequence just upstream from the coding gene itself. These protein factors bind there. We're going to talk about extensively today, but they can either be transcription activators or transcription inhibitors. These are transcription activators, meaning that when they bind to the DNA, they will actually they will signal the beginning of transcription. Okay, so there they are. I think this is actually a yeast transcription factor, not E. coli. We're going to talk about E. coli mostly today. And in a moment coming along, the RNA complex will come and find these transcription activators, and binding to them then finds the beginning of the transcription activation site. Okay, so now it's clipped onto the DNA, and this is an actual molecular picture. That kink is something that actually happens. And then there are other upstream promoters that start the initiation complex and assemble the RNA polymerase. And then there are other promoters upstream. I don't know if you guys can see my mouse, but upstream there that actually also bind to the DNA. And when the RNA polymerase initiation complex has been fully prepared, then they can activate it and initiate transcription. And it's really upstream there, so it's going to come out almost like a race car getting a tap or something, or a scorpion's tail coming along there. The activator proteins now will signal that all things are go for making a transcript. And boom, it's off to the races. As you can see, the RNA polymerase just runs along the DNA and spits out the golden chain of RNA behind it. What's really fascinating is you can see that the enzyme itself actually breaks the double helix and actually unwinds it a little bit. I think it actually breaks the DNA and reattaches it to a different configuration so that the enzyme can just run through and read it, breaking the double helix and reassembling it out the back. And polymerizing the RNA. And then at some point, it will run into something called a terminator, which is an RNA sequence that will tell the RNA it's time to stop, the RNA polymerase to basically get off the genome and stop copying. We should see that in a moment. Boom, stopped. Okay. Then after that, the RNA will migrate to a different portion of the organism. In the case of eukaryotes, or in the case of prokaryotes, it might just be they might attach to the RNA immediately right there. So let's take a look at the translation. Okay. So here's the RNA. This is a yeast nucleus, I think, or some sort of nucleus, and the RNA actually is transported off to a place called the Golgi. But in bacteria, it behaves the same way. The lower half of the ribosome, or the large ribosome unit, will attach to the RNA, and then the small one, and then the large comes along and clamps onto it, and it's sort of like the two pieces actually sort of like grab onto it like two halves of a vice. And the large ribosomal unit takes the transfer RNA, the tRNA, one by one, and matches them to the three bases that it's reading. And it has a single amino acid on the end of it here. And when the proper RNA binds to the binding site in the ribosome, that protein, that amino acid is attached to the growing protein chain. So here's a picture of, there's actually 3 places for 3 tRNA to bind to the bases of the RNA. And as they go from basically site 1 to site 2 to site 3, they leave their amino acid on the growing peptide chain. This is a gigantic molecule. It weighs 30 or 40 times as much as hemoglobin. It is huge. See, there are the RNA bases. Because RNA forms a single strand only, the bases point up and the tRNA can read them, forming basically a 3-base complex. So the ribosome can create the pep protein. There's a lot of, even though these are molecularly correct, these videos are very dramatic in the sense that this is not exactly how the peptide comes out of there. There's a lot of mystery about that, but this is essentially how it works. It's like a molecularly correct cartoon is really what it is.
Ron, there was a couple of questions in the chat.
Oh yeah, please.
One was, how do you know if a piece of DNA codes for proteins, or is it just a non-coding region?
You know, that's a really good question. And you guys are starting to become scientists. So I'm going to tell you the answer is, first answer is, yeah, we can tell that by the sequence. The other answer is we don't know. So there is software that will actually read the genome sequence, and we'll read the genome sequence, and we can look for certain sequences that we know indicate the beginning and ending of a gene, and we can guess where the genes begin and end. And if we do that, but there's an empirical way of doing it, which is basically reading all the RNA out of a sample and then sequencing just the RNA. And so those are clearly genes because the RNA has been produced. And then we put that back onto the genome and they'll say, now we know where the gene is. Obviously, the latter is a lot more reliable because it's empirically, we've actually seen an RNA transcript, so we know it's a coding region of a gene, or at least a gene. The thing about it is some genes don't turn on very often. And so you can't find every gene like that. So ultimately, the answer is we know 95% of the genes in most of these organisms from a combination of these two things. But the software still over predicts. It predicts probably 2 to 2.5 times more genes than we actually see when we look at RNA. So and then when you're talking about animal cells and other eukaryotes, the genes become very complex. We're only going to talk about bacterial genes today. That makes it super simple compared to that. You probably need a whole nother class to talk about animal genes. In any case, we can't. There's always a few genes. There's always a few little bit in the dark corner of biology and the genome. We don't know that those genes exist. Some of the sequences have never been seen, and we don't know if the genes. We can see the genes there from the software. We don't know if they're alive or dead. A lot of the genes that are mispredicted are simply just no good anymore. They don't actually show up. So there's the answer to your question. I hope that helps. Is there another question? I should probably look.
There was one more, yeah, and it asks about kind of the chromosomal level. Okay. It's just one of the chromosomes being unwrapped by a specific mRNA or RNA to transcribe. I guess it's asking is like what at the chromosomal level, how's that regulation happening?
Well, again, there's always, you're going to hear this a lot if you ask questions. There are two answers to that question. The real simple answer is in bacteria, for instance, the genome pretty much floats free, and it's always available for transcription and transcription translation. For the human genome and other animal genomes and eukaryotic genomes, it's really difficult to control and understand how the transcription works. Any given gene may be actually cut up in different ways so that different parts of the gene are actually translated to protein at different times. So if you're looking at some simple cases that are well known, we understand it very thoroughly. But there are a lot of corner cases that don't happen in the simplistic view of things, and they're still the subject of research. I hope that answered your question. So just please keep them coming. Those are great questions. So I'm going to talk briefly about what a protein is. Proteins are something that most people do not study too much because they're a lot of work. As a technician or someone who's doing sort of assisting research, you're going to see that most research is based on DNA and RNA because we can sequence it, we can handle it in very large quantities, and all the DNA and all the RNA, in one sense, they're all the same. All the RNA are long strands of nucleic acids. They may go on to become lots of different proteins, but they all kind of do the same thing roughly. Proteins are quite different. Every single one of them is an individual. This is a beta barrel protein, for instance. There are hundreds of different folds and beautiful configurations. When you look at the polypeptide, you'll see that it forms flat strands and spirally helixes, and sometimes it doesn't form either of them. And it all depends on the sequence of the amino acids. So I just, for your amusements, I put the 20 amino acids off to the left. Those are the 20 amino acids you hear about when you read the box of your breakfast cereal. You can see in the upper left-hand corner, there's a carboxylic acid and an amine on the top of all 20 of these structures. That is an amino acid. And then off this beta carbon then is a different chemical species for each of the amino acids. The chemistry off of the amino acid allows it to fulfill a practically infinite number of roles. Proteins do all the chemical work, they do all the mechanical work, they do all the chemical transformations in the cell. They are responsible, they are literally the active hands of life. And every different gene has produces a protein. The protein may act in surprising ways, many of which have not yet been discovered. Because proteins are the active portion of biology that actually do all the chemical work, they are very prized as commodities in technology. How can I move this on? There we go. So there are many, many applications for proteins. So while in academia, a lot of research doesn't involve proteins very much because they're expensive to understand, in business, the industry uses proteins for almost all of its products. So, typical therapeutic landscape here, vaccines all result in a protein. Antibodies are used to create immunity. So actually, since we're hearing so much about COVID, the president had an injection of a cocktail of a bunch of antibodies that are supposed to neutralize COVID-19. We don't really know if that cocktail really neutralizes the virus and really helps anybody get safe, but it's a pretty common way of treating lots of diseases is to actually just give an antibody infusion into the blood. And so it's not surprising that this may happen for COVID. There are lots of people who have genetic issues with their blood, for instance, diabetics, and there are people who need growth factors and other kinds of protein hormones. All of these are produced, and a physician could apply them to a patient and help their health. Clotting factors, people who are hemophiliacs, they often lack a protein in their blood that helps them form blood clots at the appropriate time and level at the appropriate intensity. All of these diseases have been cured or at least treated by protein factors produced in pharma. So if any of you goes to work with pharma and you're producing a biologic, about 70% of the treatments produced in pharma right now are proteins. They're not like things like small molecules like aspirin. They're actual proteins. It's called biologics. Okay, so a lot of industry in pharma is making proteins in large fermentation vessels and purifying them and getting them to the pure enough and formulated properly to give to a patient. Beyond that, there's another fascinating part of industry, which is rapidly growing in the job market in the Bay Area. And I've been deeply involved with these people. And this thing's called industrial biotech, right? So enzymes are proteins that do chemical transformations, and they're very, very big business. There's probably about 20 or $30 billion in enzymes produced for industrial applications. They annually, and the sales are mounting. And these new startups that are happening all over the Bay Area and the world now, they're very interested in taking that to another level. So typical applications that we could talk about are fabric care. The only reason that these detergents work so well is because even though they have very little soap, so in the 70s, it was decided that phosphorus, which is a very common part of laundry detergent, was causing red tides. And so they basically, President Nixon actually signed the Clean Water Act, which forbids people from buying high phosphate laundry detergent. And that meant it was really hard to get the clothes clean. So what they did is they replaced the detergents and the phosphates with enzymes. Instead of trying to dissolve the protein and clean it away with soap off the fabric, the enzymes go into the fabric and chew up proteins, they chew up food, and it just disappears into the water. And that's why, and so that's, you don't realize it, but you're using these enzymes every week when you go to the laundry. Dishwashers also have dishwasher detergent also has enzymes in it. There are many, many applications for cleaning machinery in industry, but also for food. So the reason, enzymes are part of the white bread revolution. Although people kind of make fun of white bread, store-bought bread keeps fresh for a week or sometimes two. If you make bread on your own in your own kitchen, it's fresh for about a day and a half. After that, it's too hard. You don't want to eat it. And that's because an enzyme has been applied to the flour in white bread to unwind all of the starch kernels in the flour to allow the baking to happen more thoroughly and to sort of like, and basically help the properties of the bread to maintain its shelf life. It's possible. 2 weeks is a tremendous amount of time for bread to stay fresh. If you've ever baked your own baguettes, you would understand this. But it's literally, enzymatic treatment of food has literally revolutionized access to nutrition in food. It's responsible for partly responsible for how we eat everywhere the same food, and we can rely on its safeness, its quality. It's the same if you go to a grocery store in Pittsburgh, Pennsylvania, or Pittsburgh, California, you'll get the same experience. And that's because all the chemical structure of the food is understood, and it's absolutely the same wherever you go. That's an amazing industrial process. And it's going to have a greater impact on other things, right? So we have, there's a lot of GMO work to improve food. Some of it's really significant. Some of it is kind of trivial. These are some interesting examples. There's the Arctic apple in the lower right. There's an enzyme that actually causes fruit browning. And all they had to do was knock it out. They just changed the DNA so the enzyme doesn't appear. And the apple can stay fresh, even if it's cut, for a couple days, right? That's kind of a trivial application. Everybody knows GMO soybeans. They're modified just so they don't require so much pesticides, or they can live through certain kinds of other pesticides. That's somewhat controversial. I think it's okay, but it doesn't change the nutritional quality of the food, I don't believe. Actually, it doesn't change the nutritional quality of the food, but it doesn't actually add anything to the quality of the food either. It doesn't make it better food to eat. People are trying to make food that actually helps reinforce your health. And one example of that is golden rice on the left-hand side. This is a rice where beta carotene the genes to create beta carotene have been added to the rice, because in Asia at the time this was conceived, it was possible people wouldn't get enough of the vitamin A, and if you have a shortage of vitamin A, you can lose your sight. So we had the rice is there to solve a major dietary issue. A bunch of activists actually got it killed in Asia. It was just that was a total bureaucratic mess. It took 20 years to straighten out the patents so that the rice could be produced. And when they tried to grow it, then the activists went in there and tried to stop it for social reasons. So that was complicated, right? On the other hand, a new generation of food products is starting to come through. These are some examples. Um, the whole concept of alternative proteins is very important right now. Upper right-hand corner is a picture about lab-grown meat. There are several companies that are trying to make meat out of the same kind of cells that you're growing in the lab right now. They turn them into muscle cells, they gather them up, and then there you got your hamburger. And it's pretty realistic hamburger. There are also companies that are trying to replace other animal-based products. Clara Foods in South San Francisco is making egg whites. And the egg whites are the same proteins that are found in egg whites, but they're not made with the chicken, right? So we're getting to the point where we can actually understand the proteins and the cells involved with the food, and we can design the food on a molecular level to make the food more healthy, to control its properties, and make them work better in the food. It'll enable the creation of new foods that don't exist. They're otherwise not possible. And it's a very exciting time. And we're going to see all kinds of new kinds of food and enhance the experience of the consumer. And then there are, I just put the whey protein isolate jar in there because, people are protein's really a consumer trend right now. If you go to GNC, it's basically a protein shop with different levels of protein quality for food, for different levels of price. Protein is a dominant factor in the food market right now. And so people are looking for lots of ways to make new foods with protein and to use some of the proteins that we know in new ways to make superior products. I just want to tell you a little bit. Most people don't quite understand how important proteins are. So I just like to put this, getting back to the biomedical topics, this is a cartoon drawn by an artist who's a professor down at UC San Diego. And what he does is he sort of looks at how much protein's in something, and he tries to accurately draw what it looks like from the protein point of view without the water. So this is blood serum. Blood serum is what happens when you take blood and you spin in a centrifuge to get out all the cells. And the serum is the liquid left on top. As you can see, it is just chock full of protein. I think it's probably more than 50% protein by weight. There's a little virus, that purple thing's a little virus in there. There's all kinds of things flowing through there. You can see all those yellow 3-lobe things are all antibodies. And there's some clotting factors and some other kind of an albumin and some other things. There's an immunohistocompatibility complex down there. So that's the thing that looks like a broom almost down the lower right-hand corner or quadrant. Everything about the body is full of proteins doing a job and making things better and reinforcing our health and our life. There's nothing dead in the body. Everything is thoroughly filled up with proteins doing your work and keeping you, basically constituting your life. Any other questions? Everybody's good? All right. So this is also a place where when you talk about microbiology, you're going to encounter lots and lots of different kinds of organisms. Normally, when we study molecular biology, you're going to look at E. coli and yeast and maybe some human cells. That is 95% of all academic research, just focus on those 3 organisms. But when you really want to understand to create a product from cells, sometimes you have to choose a cell that's capable of doing different things than normal. And the properties of the organism make a big difference in your outcome. So this is an interface where lots of exotic and fascinating biology can be learned. So these are common, these are all used in fermentation to create products. So upper left-hand corner, the famous CHO cell, Chinese hamster ovaries, one of the first maybe 6 different cell lines that existed that you can actually propagate over and over and over again. These animal cells are really popular for making antibodies and other pharmaceuticals and proteins. Algae is actually very popular for producing exotic oils and sometimes very high protein content material. Some things you can't grow in the microorganisms, and they can be grown in plants as well as in animals, but plants are a heck of a lot cheaper, and activists care a lot less about a plant that's growing one of your products. And so That's actually a common place to. Those are tobacco plants, I think, and that's the kind of plant that has that like for instance, Ebola virus vaccine is grown in, because it just doesn't grow in microorganisms very well. You can actually culture plant cells separately. I'm just going down the list, and you can even propagate them into whole plants. So if you want to make plants with special properties, you can do genetics on them and see how they grow as plants. There's some muscle cells, like the kind we use to grow lab-grown meat. The lower right-hand corner is one of my favorites, fungi. So yeast are a kind of fungi, but they tend to grow just in terms of, just as sort of round little cells. Most fungi form hyphae. So what you see here is a network of the mycelial hyphae that are growing out from the cells. It's a fascinating network. So each of those hyphae have multiple divisions in them. So you just imagine a tube with little walls down the tube. That's kind of what happens to the fungal cells. They are completely crazy things where the nuclei will travel up and down the hyphae, passing the cell walls, and they will just migrate around the organism to wherever they want to go. So the nuclei roam freely through the hyphal network. And at the same time, these things are capable of producing a lot of protein. They are possibly the world champs at producing proteins from other organisms. And so they're very much favored by industrial biotech. And I've used them as well for that purpose. Okay, any questions? Okay, now we're gonna start, cell-free biomanufacturing. Okay, yes, let's talk about that really quickly. So, in a eukaryote, you've got the nucleus where you've got the DNA and you've got the RNA polymerase, and then the RNA comes out, and then the ribosome comes on, creates protein. It's got to move to the Golgi apparatus or one of these other organelles for the protein to come out. It's kind of complicated. And so what someone decided to do is like, let's just isolate the inside of the cell, the fraction that's related to translation and transcription. And let's just make a liquid out of that. And so basically, if you just spin down all the nuclei with the Golgi and you sort of break open the membranes and you take the insides and you just make a test tube of it, that is basically a cell-free manufacturing solution. And you can put your DNA in there and it will just start making protein. So it's a lot of work to produce that stuff. I think it's 2 or 3 days of work to produce a cell-free nuclear-free prep for that. People have scaled that up, and you can do that for some things. It's really good for screening some things, but it's fairly expensive to produce protein that way. And to my knowledge, I don't think there are any products that are produced that way, but it's a great research tool. And I know there's some interest of us doing some work eventually at Laney with those things. And so that's a really great question. Okay, does that answer your question, Nathan? Okay, I'm going to assume that.
Oh, thanks.
Great. All right. So you guys may have covered this. I'm just going to go fast and just cover it all again real quick so it's fresh in our minds. So these are the 3 branches of life. There actually is a 4th one, but you could just put 2 copies of the prokaryote up there, and basically that would be it. There's a couple kinds of bacteria. They're prokaryotes. The other kind of bacteria. What is the other kind of bacteria, Doug? It's the eubacteria.
Archaeans?
There's eubacteria and archaea. So prokaryotes are eubacteria, and archaea bacteria. They have, for a long time, the bacteria were clustered together. Then someone realized that the ribosome is substantially different in the archaeobacteria. So we kind of have 2 kinds of bacteria, but they physiologically, their pictures would look very similar. All bacteria are just kind of bags of protoplasm where there's a nucleus and a bunch of protein and some water in there, encapsulated by some kind of membrane. And that is the easiest thing to work with, because since all of the proteins are we'll actually see some examples of that today, but bacteria are just a heck of a lot easier to work with. They are the best way to they're the workhorses of producing DNA. that we work with. And you kind of have to work with bacteria almost no matter what you do. On the left-hand side is a eukaryotic cell. I think it's a generic one. There's a nucleus in the middle, and there's a bunch of other organoids by the nucleus. There's the blue or mitochondria. There's a pink Golgi apparatus. There's a so-called rough endoplasmic reticulum. And then there's vacuoles and lots of other little compartments inside the cell. The eukaryotic cell is almost like a body unto itself, and it's very complicated. And each of these organoids is basically an organ, basically got a membrane and a structure of its own. And it's a much more sophisticated organism, and actually it's much harder to understand how it works. But on the other hand, we are eukaryotes, and we have very complicated cells. And so they're studied partly because that's where human beings are. And it's probably 90% of the complexity and 10% of the biomass of the world is eukaryotes. And on the right-hand side, you can see a virus. Virus that is not a cell, it's just an envelope. We all have learned a lot about viruses this year, but it's an envelope that contains some form of nucleic acids, could be DNA, could be RNA, could be multiple pieces of RNA, encapsulated inside a protein coat, which might be a lipid coat or a membrane coat, as we say, that looks a little like a coronavirus. A lot of viruses look like that. They just have one or two kinds of protein on the surface and then a bunch of RNA inside. They're not very complicated, but they sure can cause trouble. Okay. We are now getting onto practical. How are we doing for time? We probably 40 minutes in. Okay. All right. So that's great. We have lots of time for you to do your hands-on practice. Okay. So the rest of the talk pretty much is going to focus on plasmids. And it's not because this is basically the most straightforward system to learn from. It is the simplest genetic system to work from. In bacteria, for various reasons, the chromosome is not enough. And so bacteria kind of keep these little bits of DNA around, and they just hang on to them almost like trading cards. They can get rid of they can decide to throw away the plasmid, eject it out of their cell. They can find other plasmids and bring them inside. And the genes on these plasmids then become active in the bacteria and may actually give the bacteria some advantage in its life in terms of competing for food or what have you. So because bacteria are so relaxed in terms of how they treat their genetic material, that has turned into a really nice system. There were actually researchers here in Berkeley at the University of California who discovered the first plasmids and started to break down the DNA in terms of its function so they could understand how to use the plasmid as basically a tool bench for putting genes inside bacteria to see how they would work. It was a major change in the way biology behaved and a major step in the development of modern molecular biology. Okay, so lower right-hand corner, you can see this is a plasmid diagram I just pulled up randomly from the internet, but all of them have pretty much the same sort of structure. In fact, this resembles very much resembles pGlow, which we'll be looking at later. But I think rather than explain this diagram, we're going to move on and look at some other examples, and you'll be able to pull up SnapGene at the same time. So I would like everybody, therefore, to pull up SnapGene and get the pglow.dna file out so you can follow along with the diagram on your own. I'll give you a couple minutes to do this. Actually, you know what? It's been about it's almost halfway through. So I want everybody to do that, and then we'll take should we take a 10-minute break for bio, and then we'll come back and do the last half of the class?
That sounds good.
Okay, so you guys have a few minutes to sort of find the file. If you need to install, you can install now, and we'll come back up with the pGlow file in a few minutes.
Okay.
Okay, yeah, all right, sorry, we'll start now. So just a note about circular versus uncircular. There are non-circular plasmids. And so with this switch view that Doug mentioned, it basically will cut it at the so-called origin and just lay it out flat. And to be honest, it's really hard to work with a circular picture. And so a lot of people actually prefer to work in the straight view. We just have to remember is that it really is circular, and so the little dots indicate that it should be able to come back.
I did the hack that the student who I forget her name just mentioned, and it worked for me as
Great.
well.
Good. Okay, so I guess hopefully everybody can see it circular, but it's really not that big a deal because, you know, in my mind, I look at it linearly almost all the time anyway, because it's just so much easier to work with linear. But we will get on with it now. So I'm gonna turn my presentation back on. Let's talk a little about this. Well, maybe I'll just do desktop. No, it'll get covered by the the Zoom. All right, so let's talk about pGlow. So first of all, what is pGlow? pGlow is actually a plasmid made by Bio-Rad for instructional purposes. I've drawn this really horrible diagram by hand. You have a nicer one in front of you. There's multiple things on the pGlow plasmid, but in fact, what it does is it produces a protein called green fluorescent protein. Green fluorescent protein was discovered in the early 90s, and it comes from a jellyfish, which if you look at jellyfish just right, they do fluoresce when ultraviolet light hits them, and they give off this green glow. They actually give off light. And they found the protein that actually fluoresces. It requires, it fluoresces in the sense that light comes in at a higher frequency and leaves at this green frequency. And when you put pGlow into bacteria under UV light, you can actually see the GFP fluorescing. And that's what the plates are at the bottom. It's an incredibly useful protein because one protein to our eyes, one protein in the cell looks very much like the others. And the fact that GFP actually is something you can see very easily has revolutionized molecular biology to an amazing extent. It's one of the most popular proteins, and it's used to test all kinds of things. Okay, so I think, are we going to actually cover? Yeah, you know, I'm going to cover GFP in a minute. I'm going to go back and I'm going to cover PCR now. We're going to talk about how to manipulate a plasmid. And I think you guys may have seen this before. We're going to do it again.
Polymerase chain reaction, or
I'm going to
PCR,
turn off the,
uses repeated cycles of heating and cooling to make many
all right, so I'm turning this down. So as you heat up DNA, it actually starts to break up, and the double strand just gets so hot that it falls apart along the entire length, somewhere around 85 to 95 degrees Celsius. It pops apart. And then you can make these small things called oligos or oligomers of short sequences of DNA. And when the DNA is apart, the oligos can come and bind to the reverse complement that they're designed to hit. And then we have some DNA polymerase in the mixture, and it will actually run off and turn the oligo and try to create a double-stranded DNA from that. And we repeat these cycles of heating and annealing, which is to say the oligos attach, and the DNA polymerase then does another round. We can do this over and over again. Usually we do between 20 and 50 times. Every time the number of DNA, the amount of DNA in the mixture doubles. Now you can see that this short segments are going to become more and more frequent because basically we have two different oligos, one at each end of the desired sequence. And so at the end, after about 20 steps of that, we have a tremendous amount of the oligos of the DNA that's desired, right? If you look at this diagram, this is a diagram of the movie we just saw. Over time, for every cycle, there's more and more of the chains that are just as long as we wanted, beginning and ending with the two oligos that we designed to clone the DNA out of the originating DNA sequence. Is everybody good with that? Any questions? Doug, I can't do full screen and see the chat, so I'm gonna kind of ask for you to be in charge of the chat.
I got it.
Thanks. Okay, so that's one of the major tools we're going to be working with today is PCR. This is another thing that originated in the early 90s, and boy, did it change everything. Then there's the restriction enzymes. This is a very important tool. It's ubiquitous. Everybody knows about them. They're used less and less for reasons that we'll discuss a little later in the class today, but they're super important. These actually, bacteria don't have an immune system, but they have lots of different proteins that help them fight off infection. And one of them is the restriction enzyme. So almost every bacteria has a restriction enzyme, and the restriction enzymes are slightly different. They bind to very specific sequences in double-stranded DNA between, let's say, 4 to 6 bases, 4 to 8 bases long. And they will cut the DNA in both directions. So at the top, you can see on the right 5 different restriction enzymes discussed. The top 2 are blunt cutters. They cut the DNA exactly in the same place on both strands, and then just the blunt ends, as say the ends are pretty much just flat. They just sort of fly free. ALU1 is probably an enzyme from an organism that doesn't actually have this 4 DNA bases in its genome. And so any virus that comes in that happens to have those, it'll cut the DNA up, right? So that's pretty obvious how that's helpful. BAMH1 and HinD1 and EcoR1 are 3 of the most common restriction enzymes. I think EcoR1 comes from E. coli, in fact. That's how common it is. And those guys cut the bases, but not in the same place on each strand. And interestingly, if you look at all these 5 sequences, you'll see they're all palindromes. So if you read BAMH1, it's GGATCC on the top. Reading from right to left on the bottom, it's GGATCC. It's exactly the same sequence, but reverse complemented. So the fact that these guys use palindromes makes it really easy to spot them in the chromosome, and they're very specific. Even one base change and the enzyme won't cut. And then they leave these 4 loose bases between them. Okay, those are called sticky ends in the business. I also put BGL1 there at the bottom there, and sometimes it'll cut when there's where there's the 6 specific bases are separated by bases. So all kinds of exotic species of restriction enzymes are known, and some of the companies make hundreds of different of these things, and they are somewhat expensive, like $80 per to buy some enzymes. Okay, so how plasmids are have been created up to now. When I mean now, I mean up to about 10 years ago, is that people use these restriction enzymes to cut pieces out of the organism. So when we go back to pGlow in a minute, we'll find some spots where there's lots of restriction enzyme sites, and you can buy the restriction enzyme and cut them out. On the right-hand side, you can see you can cut with these 2 enzymes, E and S, and cut this part out. And then you can cut the plasmid on the right with E and X. And then you can create, this is a system at IGM to sort of create these 2 sequences right next to each other. It's a system for putting parts together. And so the restriction enzymes help it become extremely orderly to sort of build things up and so forth. But the problem is when those parts are being inserted, they only are held together by 4 bases, 4 sticky bases on either end. And then you have to apply another enzyme called ligase. And ligase will actually put everything together. But 4 bases on the sticky end do not stick very much. And so way over 95% of the DNA in the mixture never ligates. And you're lucky to get just a few working. And if you are a little sloppy or not super careful, this reaction often fails, right? So this is the main skill of a molecular biologist from like the 70s through the 90s, was to be able to make this stupid reaction work. And it's not easy. You really have to have experience and talent to do it. Once you've got your protein in the plasmid in such a place that the protein will come out, it works really well. So this is just a block diagram of the process. We're just going through the process of working with a plasmid step by step here. You get a few colonies out after you've done your ligation and your enzymatic cuts and so forth. Then you can take those and put them in a bioreactor, a shake flask, or the bioreactor like the one you see below, or the one that's in the corner of the classroom. And we can cut open the cells after the cells grow. And you can see on the right-hand side, that's a SDS page gel. Have you guys covered SDS page gels? Okay, so we've been through agarose gels, right? Yes? Yeah, Doug's saying yes. So I'm believing Doug. So SDS page gel is basically a gel for proteins instead of DNA. And so this gel on the upper right-hand corner is showing first a set of molecular weight standards, because proteins vary roughly between 15 and 100,000 Daltons, 15,000 and 100,000 Daltons. Dalton being a unit of molecular weight. And you can see that the plasmid in this case has produced a protein of approximately 25,000 Daltons, and it produces a lot of it. And so once you've got a protein that's dominant in the mixture like that, you can usually separate it out and clean it up, and you can basically get pure protein from a system like this, right? That's the technical process from soup to nuts. Does anybody have any questions? Is everybody okay with this? Yeah, see some nods. Please feel free to let me and or Doug notify, or just turn your mic on and pop up the question. It's perfectly fine. Okay. I just put this slide to talk about some other technologies that I find fascinating. There are many more of them, but in terms of getting, other than using plasmids, you can, these are some other things you can do. So upper left-hand corner is editing the genome. There's more than one technique for editing the genome. CRISPR-Cas has definitely got a lot of advantages to it. We won't be covering CRISPR-Cas today. That might be in a later talk. But basically CRISPR-Cas is a bunch of enzymes that help you edit the genome and put in genes or take genes or DNA in or out of the genome as you like. It's extremely versatile. It works really well in the lab. It's a lot harder to get it to work in living being. Upper right-hand corner is a piece of plant technology. This agrobacterium transformation works amazingly. It doesn't work very well, but it's amazing things. It's a lot of work. So that it turns out that when you look at a fungus that's attacking a tree, sometimes the fungus is actually transforming the DNA of the tree and making all these galls that you see. And so in one case, the agrobacterium was isolated, which had this plasmid called TI. And it's a really complicated plasmid with many proteins in it. And these proteins form a complex. And the agrobacterium will actually inject the protein DNA complex from the agrobacterium into a plant cell, through the cell wall, through the woody cell wall. And once the T complex goes in there, it will insert the agrobacterium gene randomly at a random place in the tree genome. And that's pretty freaking fabulous, because all these other genome editing sites that we know are not, they are position specific. And so this is a completely different way of doing things, and biology's created this tool, and it's used on a lot of things that are not plants now. But it's just another sideshow on the highway. Viral transfection. So when you're working with animal and plants, animal cells, viral transfection and transient transfection are very popular because animal cells are very delicate, and what doesn't kill them often won't transform them. So viral transfection, actually, viruses edit the genome themselves. So you can actually make a viral particle. Instead of putting the viral genome in there, you can put your own gene in there, or you can manipulate the viral genome so that it adds genes as you like. That's a popular way of putting a gene in an animal cell. Transient transfection is even simpler. You can actually take a bunch of DNA and wrap them up in a sort of a lipid bilayer, and those things will actually be brought into the cell. And even though they don't integrate into the DNA will actually create protein from it, and for a few hours or a few days. And so it's kind of funny that you've got these really complicated, very sophisticated animal cells, but they'll just suck up the DNA and make the proteins on there, just like opening an email from a stranger. Okay, everybody good now? All right, so now we're going to go back to SnapGene, which you've all got open, and we're going to look together at the genes on the plasmid. This is actually a copy, a screenshot from SnapGene. So if you don't get it working, it's perfectly fine. So I've turned off the, what most of you see the different on your screen versus my slide is that there's a lot of restriction enzyme cuts being labeled. I just turned them off because I don't care about restriction enzyme cuts anymore. But that, since it's such a common tool, the people who look at DNA with SnapGene want to know where all the restriction enzymes cut. You can turn that off on the upper left-hand side with a button if you want. We will talk about restriction enzyme a little bit, but not too much. Fortunately, cloning no longer requires us to care about what restriction enzymes are in there. It's really difficult. So if you want to put a gene in place of GFP in pGLO, for instance, there actually are places to do that, to cut the GFP out and to insert the other gene. But when you cut that gene, it's really, sometimes you have to do a search through your new gene to make sure that the gene that the site that you're cutting GFP with won't actually also cut your gene of interest. And so you got to keep track of a lot of stuff using the old method. In addition, the fact doesn't work very well. Anyway, so looking at the plasmid, you can see that there are more than one thing on here. So in the lower right-hand quadrant, you can see GFP. That is the gene which we want to express the protein from. But yet there are all these other things that are required to make it work. Okay. Just briefly, I'm going to go over the origin. Upper left-hand corner is ORI, the origin replication. So this is what the group over in Berkeley actually discovered, was that one segment of the plasmid was necessary for the bacteria to copy the plasmid. So once the plasmid goes into E. coli, it's copied many times. In some strains of E. coli, there'll be 200 or 300 copies of the plasmid sitting inside every cell. And how often the copies are made is dependent upon the origin of replication, its sequence, right? So it's a signal that basically comes along and says, DNA polymerase, copy me. There are two origins here. I'm actually not sure where the other origin is. Generally, this is only stored in E. coli, so it might just be that there's one long origin broken into two pieces, or there might be a second origin just for the heck of it. There are lots of plasmid with more than one origin replication for different organisms, though. I don't think pGLO is like that. Lower left-hand corner is the BLA protein. That is ampicillin resistance. So when the plasmid, so whenever you do a transformation, nothing is complete. So when we have a pGLO plasmid or some other plasmid, we can mix a little bit of it with a bunch of prepared bacteria. These so-called competent bacteria will then suck up the plasmid, and then we can have our new cells. However, transformation is not 100%. In fact, it's probably more like 0.01%. And so we need to have a way to get out only the cells that have the plasmid of interest. In the case where it's GFP, you can just see if it's green and pull it out. But that GFP's only characteristic is that you can see it. All other genes don't have that characteristic. And so in general, we want to be able to get rid of all of the cells that are not infected with the plasmid. And so the plasmid has this ampicillin resistance gene, which is another protein that literally makes the cell live when it's in touch with, in this case, ampicillin. Okay, so we actually make a plate with the ampicillin in it. I think you guys have seen this. And we spread all the bacteria out on the plate, and all the ones that don't have the plasmid just simply die. And that makes it very easy to recover the cells that contain the plasmid. So that is called the selection marker. And selection is really important because if you can't get something back from your work, then there's no point in doing your work in the first place. Upper right-hand corner is the arabinose repressor, ARA-C. That is very interesting, and that will be the subject of our next discussions. So actually, I have done here what the gentleman in the class already did so carefully. I've just decided I only want to look at part of this, and I want to look at flat. So the BLA, the ORI, and the arabinose repressor genes, I'm going to call those the operating system. And the program, if you will, for the plasmid is just this quarter of the plasmid, right? So when we manipulate the plasmid, basically we're just looking at this gene insertion site. And there are 3 parts to every gene insertion site that we really have to take care of. There is the promoter. So the promoter is the region of the DNA where the activators bind that we saw in the movie earlier. Without that, the RNA will never be copied. Whatever's next to the promoter is copied to RNA. If you don't have that, or if it's messed up, it simply won't work, right? But it's an important part of what we're doing here. In this case, ARA-C binds to that promoter in some case. I think we're going to go over that in detail in a little bit. But if we're just processing it, figuring out where should we put our gene in and how do we manipulate the plasmid, then we look at the multiple cloning sites. So again, back from the days when everybody used restriction enzymes to cut everything, they actually would make a so-called multiple cloning site in these plasmids where there'd be like 8 or 10 enzyme location binding sites or cutting sites in a row. And we would insert the GFP or our gene of interest between those, somewhere in that set of restriction enzyme sites called the multiple cloning site. It's still a very important site because it tells us where to put the gene that we care about. And of course, GFP in this case is a gene that's already inserted into the plasmid. pGlow can be used for lots of things, including putting a protein interest right next to GFP so that the protein, your protein of interest comes out and it's also glowing green at the same time. You just attach your gene of interest to GFP, and then you can get so-called a tag protein like that. Lots of ways to use GFP. So just remember, this is the operational end of the plasmid. It's where we touch, everything else is important, but we never touch it because if any of that stuff is disturbed, the plasmid will not work, right? So this is the only part that we actually get to touch. Okay, now I'm gonna talk about ARA-C. This is the arabinose operon, the first set of genes for the class today. So on the bottom is a segment of it's actually a piece of a chromosome of a bacteria that can process the sugar arabinose. Arabinose is just another sugar. It's a lot like glucose, but their atoms are in slightly different place. And in order to utilize arabinose for energy, the ARA locus has to be activated. So there are 4 genes here encoding for 4 different proteins. ARA A, B, and D are enzymes that break arabinose down and rearrange it for use by the bacteria for energy. ARA-C is the regulon, is the transcription inhibitor activator protein, or the regulator protein for arabinose. So there are 2 promoter regions. There's PC and PBAD. You can see PBAD is just called because it activates ARA-B, ARA-A, and ARA-D in that order. And And then PC is actually a different transcriptional activator that will transcribe ARSC, and they behave very differently. So PBAD is called a tight promoter. It basically is either fully on or fully off. You will not see an intermediate amount of BAD RNA produced. PC is so-called a leaky promoter. So ARSC attaches to it, and when it does, there's less of it, it turns the gene down, but there's always, if there's no ARSC in the cell, the promoter promotes a very small amount of ARSC to be produced. So the whole purpose of the PC promoter is to turn it off when there's enough ARSC around, but otherwise just a little bit's going to be made because we always need a little bit of it in the cell at a time. When arabinose shows up in the feedstock of the bacteria, the arabinose binds to ARSC, and when it binds to ARSC, it binds to PBAD, or I'm sorry, it detaches from PBAD. So ARSC binds to PC and PBAD, but then when the arabinose is around, actually my diagrams got lost that, but when the sugar binds to ARSC, it actually kicks off the genome, and then PBAD gets transcribed. Okay, so that's kind of how it works. Interesting, ARSC is both an inhibitor and promoter. It just depends on what promoter it's bound to, and it also binds ARSC, so it's a sensor. It binds arabinose. It's a very sophisticated system, and yet it is incredibly simple compared to how yeast and mammals work. But this is a typical bacterial regulon. Okay. Is everybody clear on that? That was a lot. Is everybody happy with that? Okay. We could spend all hour just talking about arabinose metabolism. I know you'd be excited about that, so we're gonna keep going. So, swapping between the two slides, I just replaced BAD with GFP here. And it's the same thing. They just copied all the DNA from the ERA control system, including ARSC, PC, and PBAD, and then they just stomped another protein in place of PBAD, because nobody cares if arabinose is right. So in order to get pGLO to work, you just put a little of arabinose into the in there, and the protein transcription turns on, right? So that's how the plasmid is made. We're actually copying a little bit of genome function into the plasmid and just letting it go, and it behaves the same way. We made a machine. Awesome, right? It's not so hard. We'll get into more details. There's always another level. Okay, everybody happy with this?
Wait, can I ask a question real quick?
Yes.
So the arabinose connects to the PC.
I can't really hear you.
Turns on.
Sorry.
Oh, sorry.
Try again.
Is this better at all? I don't know.
Try again.
Okay. Well, I was gonna say, so is the arabinose connects onto the PC thing that makes the ARSC that turns on the other one. Is that how it works?
Yeah, let me go over that again more carefully, because I want to. So first of all, there's a sugar called arabinose, and unfortunately, it looks like my picture of the sugar got cut out of this. But the sugar is not a protein, and it's not on the diagram. But all of this stuff is called. Okay, all this stuff is called ERA because it's related to arabinose. That's how geneticists think. And so the ARSC is actually a protein that binds to the DNA. It's called a transcription regulator. Okay, and the uppercase ERA, A, means that it's a protein that comes from the gene. And the lowercase a means that it's actually a gene in the DNA. So that's how we sort of keep the nomenclature straight. What's missing here is that when the ARSC gene makes ARSC the protein, uppercase ARSC, that protein will then bind to PC and PBAD and execute its function of sort of turning things off. And then what unfortunately the diagram needs is a little purple sugar to come along, bind ARSC on PBAD, and it will kick it off, and then it will kick it off. And so that's why there's an ARSC floating above, That's actually what happens once arabinose, the sugar, shows up inside the bacterium and RSE binds it and decides to take a hike, basically. And that's what allows the RNA to be produced. So hopefully that was more clear. Are you okay with that?
Well, how come the RSC gene gets turned on by the RSC product? It seems like it's sort of circular or something.
Well, PC and PBAT are different sequences, and literally the behavior is very different. Like there are actually lots of regulators that are both inhibitors and positive regulators. They activate the gene, or they repress, or they sometimes do both. Biology is a place where, especially the genome and the proteins, there are no really strong rules, and you'll often find lots of contradictions to terms like that. So RSC, that's kind of why I like this, because RSC has different effects in different circumstances.
Okay, sorry, one more question.
Sure.
So wait, so this gene, the RSC gene, is turned on by the same thing that it produces, or am I confused still?
It's actually turned off, but it doesn't matter. Like turning on is just a reverse of turning off, right? So you'll find proteins that do all these things. Now remember, it doesn't produce arabinose. The ARA genes actually start to consume arabinose. They're actually metabolism genes that allow the bacterium to utilize arabinose for food. Does that make sense? So the arabinose shows up, and then the RSC activates all the ARA genes, and then it consumes the arabinose, and then eventually the RSC will bind back to PBAD and turn off the genes, in this case.
Okay.
Yeah? Okay. Well, let me know. I think it's probably still bothering me, but I think the logic is there, but it's kind of twisty. Okay, well, let me know. I'm just going to talk a little about genes. Let's talk about genes beyond bacteria and some terminology. So, the diagram, the line is the chromosome. The blocks, or sometimes the arrows, are the coding sequence. That's the portion of the DNA that will be copied to RNA, and the portion of the RNA that will eventually be translated to protein. There are untranslated regions on both sides of the CDS, which are sometimes called 5' and 3' UTR, or upstream and downstream UTR. They contain regulatory sequences that turn things on and off, and they stop and start transcription. In eukaryotes, not in bacteria, the RNA can be cut. So parts of the RNA can actually be spliced out and rejoined to turn an RNA into a smaller RNA that eventually gets translated into protein. This splicing can happen multiple ways for some genes, and we're not going to talk about it too much because we're not going to have any splicing in the plasmids because they're bacterial. But I just want you to be aware that in eukaryotic genes, the genes can be very long, and a lot of the RNA can get cut out. Like sometimes 95% of the RNA can get cut out before it's translated to a protein. There are two other things. There are three other elements within the coding region, or just next to the coding region, I want you to understand. So the coding region may have this, basically, once the RNA is transcribed, the ribosome has to bind to a ribosome binding site. That's the first site that we saw in the first video, where the little proteins are hanging out there waiting for, that's where the RNA binds in the second video we saw. So the ribosome will bind, once the RNA is available, the ribosome will seek the ribosome binding site. And it will also, part of that includes a start codon. So the beginning of every gene starts with an ATG, which codes for an amino acid called methionine. This is why it's so difficult to detect a gene, though, because methionine is found throughout the coding region. It's found all over the place. So the composition of the ribosome binding site and the start codon somehow create ribosome starting of ribosome translation. The ribosome comes off at something at the very end. There are 3 codons that actually indicate the stopping of the gene and translation, and those are called stop codons. So the beginning and ending of the gene are fairly easy to find, unless they're not. But those sequences are fairly canonical. They're fairly much the same in every case, like 95%, 98% of the cases. They're easy to read. And you'll see in a minute that SnapGene helps you read those things. And then beyond the end of the stop codon is a terminator. So I should have put that on here. Those arrows are supposed to be terminators, those little gray arrows. But a terminator tells the RNA polymerase to stop polymerizing. So it stops copying the DNA at a signal that's outside of and past the end of the coding region of the gene. But after the coding region is fully transcribed, you don't want to make any waste RNA. So the untranslated the terminator will tell the polymerase to stop polymerizing. Okay. Okay. Now we're going to go through and design a plasmid for another purpose. Okay. So this is example 2. You may now pull up the PET. If you want to look at this in detail on your own SnapGenes, you can pull up the PET GFP DNA file. I'll give you a couple minutes to do that. Okay. Is everybody ready? Well, say something if you're having trouble. I'm just gonna talk at this slide for a couple of minutes. So PET is your second plasmid, and it showed up in the 90s, and it is the champion bacterial protein maker. It's just the best. This is PET without EGFP in it. It's very similar to the map that you have though. As you can see, it's very similar. It's very similar to the other plasmids we looked at. They're based on, all these plasmids are based on the same plasmids that came out of Berkeley in the 1970s. They actually are almost exactly the same sequences they were in the 70s on the left-hand side there. So as you can see, there's 2 origins again, right, necessary for replication. And then there's a selection marker. It's in a different place, but it's a kanamycin marker. So there's actually 5 or 6 different antibiotics in use. And that allows us to actually have more than one plasmid in a bacterium at once. If you can actually put, I think, up to 4 plasmids with different antibiotic markers in them. It's really hard. The more plasmids in a bacterium, the harder it is for the bacterium to hold on to them all. But in a pinch for a short experiment, you can get multiple ones in there. So there are 4 or 5 different antibiotics you can use. Kanamycin and ampicillin are the most 2 common. And then what they decided to do is, instead of using the old promoters, which would produce a few milligrams of protein per liter, they decided to use the strongest RNA polymerase promoter they know of, which is from T7 phage. So T7 phage is a micrograph there and a little artist rendition of the T7 phage, which looks like an alien spaceship. It's an amazingly complicated protein machine. So this is a bacterial virus, and I don't know why bacterial viruses are so complicated compared to human viruses. But as you can see, there are multiple parts. Each one of the parts, the tail fibers, the base plate, the core, the collar, and the head, they all have their different proteins. I think there's like 13 or 20 proteins. I don't even know how many are in the T7 phage. And this thing has enough DNA in it to code for these proteins. And once it injects, it actually, the base plate somehow injects the DNA from the head into the bacterium. And then the RNA polymerase and everything go wild. And then the bacterium basically bursts open full of phage. So it's a really effective system for killing bacteria, but it also means that these genes are transcribed and translated at a tremendous rate, much, much more than any gene inside E. coli normally. So the people thought, wow, if we want to transcribe and translate a gene in bacteria, we should use T7 instead of, people were using things called things like LAC and all these other genes that were known from E. coli genetics. And these guys put this together and it was an amazing success. It typically produces 20 or 30 times more protein. And that's what we're going to be playing with today a little bit. So as you can see, PET has this extra gene here. On the right-hand side is the LAC inhibitor gene, which is very similar to ARA-C. And that's an old gene that also is a very common promoter for plasmids. But they basically got this LAC to activate the T7 polymerase. So there's a T7 LAC promoter in front of the black arrow. The black arrow is where the gene would be put in. That's the multiple cloning site. And then there's a T7 terminator that you can't see, but you can see it on your SnapGene readout. And so how this thing works is they made a special bacterium that has T7 polymerase, RNA polymerase in it, in this genome. And the T7 polymerase is sitting around in the bacterium ready to go. And this plasmid gets activated by you adding, there's a chemical called IPTG, which binds to LAC and gets a transcription going. So, but instead of getting the regular E. coli RNA polymerase on there, this LAC gets T7 polymerase going, and it just makes lots and lots of copies of the RNA. In addition to which, BL21DE3, the bacterium that is supposed to be used with PET, BL21DE3 has 2 proteases removed. So it basically, they just took the proteases out to improve the amount of protein produced. So this is a primitive strain to be used for protein production. There are many, many other strains to produce protein in very large quantity. It's a very sophisticated science, but there's your example for today on how to improve your protein yield from a plasmid. And that's all been done for us. We have BL21DE3 here at Laney, and we have PET, and we're going to start working with it. What we want to do is we want to make a super bioluminescent bacterium. So right now there is a plasmid called PJE202, which is the third plasmid that you have for SnapGene, and you're welcome to load it up and take a look at it along with me as we talk through the slides. So your third genetic pathway I'm going to talk about is quorum sensing and bioluminescence. So in the upper right-hand corner, this is the Lux system of promoters, okay? So what we're going to do today is we're going to put Lux in T7. That's just to tell you why we're going through all this rigmarole. It's a very long story. So, through a lot of effort, a bunch of researchers discovered how so-called quorum sensing works. There's a bacteria called Vibrio fischeri. There's actually quite a few bacteria that do the same thing. But if you grow Vibrio fischeri together in a culture, when there's a certain density of bacteria, they start to glow. And so there's this weird social behavior the bacteria have. And I don't think that we actually understand why the bacteria glow. I don't think they have any light sensing capabilities, but they do it anyway. And what happens is that there's a gene called LuxR and that activates, I think, 6 other genes. Actually, you can see it there in your PJT02 file. One of the genes synthesized and called homoserine lactone, which is called AHAL here. And so every bacterium is always putting out a little AHL. And when the density becomes very high, and there's a lot of bacteria in a certain volume, they start sensing, hey, there's a really high concentration of AHL here. When we have a high enough concentration of AHL, the homoserine lactone binds to LuxR, and then it starts copying the genes. And then the genes then create bioluminescence. Okay? Has everybody got that? That was a mouthful. I hope the diagram helps. The bent arrow is actually, the bent arrow here is actually the promoter region. This is where LuxR binds to sort of regulate the transcription. Okay? And you can see there's an RBS and ribosome binding site and a terminator here. And then there's another set of genes to the right. Yeah? Okay. So on the left-hand side of this diagram, this is some heavy science here, so we'll just take this as it comes. But I'm not even explaining too much, but basically the researchers cut a bunch of genome sequences out of Vibrio fischeri, and they found this section of the genome contained everything you needed to make E. coli light up when they put it in there. And so we actually were able to obtain this plasmid because the Odin sells it, and it contains all these genes, LuxR through E, so 7 genes. So if we look at the diagram on the right and the diagram on the left, you can see LuxR it goes in a different direction, and the RNA, it's actually got 2 RNA coming off of it. And LuxR, it basically, it's just like ERA in the sense that LuxR sort of makes a small amount of itself. It sits here and turns off transcription of LuxI until there's enough homoserine lactone. And then boom, it starts producing the longer RNA. Okay? We'll go over that again in a minute. There it is, a little clearer. Okay, so just to show you how this works on a protein level, there is a bidirectional promoter between LuxI and LuxR. That's why the arrows will look all tangly. And there's terminators on either end, okay? So as you can see on the right-hand side, there's going to be an RNA produced with 7 open reading frames in it. 7 coding regions will be in there. The one on the left only has 1. This is called a polycistronic RNA, and they're not found in eukaryotes. They're only found in bacteria. Bacteria are so reasonable, and eukaryotes are so mysterious. But basically, this is a really easy way for the bacteria to control the proportions of the different genes. Like, because there's only 1 strand for the whole thing, basically the proportions of every gene, A, B, C, D, E, G, and I, will all be kind of like what you want. There won't be any waste protein. There'll just be enough to be made, right? That you need, right? And so I've taken the liberty of attaching a red circle for the ribosome binding site to where the ribosome's gonna make 7 different polypeptides from the right-hand RNA, but the RNA is going to be pretty big, right? And so what we want to do, and then you can see by the color, I try to match the colors on the long RNA to the 3 proteins that make light. So there's 1 gray protein I didn't put in. LuxI actually creates homoserine lactone. But LuxG is a flavin reductase, and that cycles this flavin mononucleotide and reduces it. The reduced form then is, and then there's a fatty acid reductase complex that actually takes fatty acids and turns them into aldehydes. So the ROH molecule up there is called an aldehyde, and it has a lot of, it's actually very reactive, which means it stores energy. So the Lux luciferase is genes A and B, it's a dimer. Those 2 proteins get together and they can turn reduced flavin and the aldehydes into light. Right? So this is great. All these genes are here together. This is all the functionality you need to make light in any organism, in any bacteria, right? And then the fatty acid reductase complex actually is the product of three genes. It's a C, D, and E, I think. right? It seems like a lot of work. Thank goodness we don't have to do it ourselves. The cells do it without thinking. Yeah. Is everybody okay with this? We don't really have to remember exactly how this works. We just remember there's a lot of genes here, and they're all necessary to make light. Okay. So this is what we want to do. So the team was sitting around, and we actually have a collaboration with a YouTuber influencer, and she wants to make a vial of bacteria that glows super bright. And when you guys, does everybody get to play with PJE202? Is that a lab here?
Yeah, so our Bio75 class has already done this experiment at home.
So some of you already got to see this, right? Does everybody in this class, has they seen the results? Maybe not. Anyway, so if you put PJE202 into that and you grow it to super high density, you have to turn off you have to take it in the closet and turn it off, and then you can see a blue glow. Like, we made some of the densest cultures of PJE202 possible, and it's like 4 times brighter that way. And you have to turn off the light and keep it off for a long time to see it. It's just glowing very faintly. That's how much natively you expect, okay? We would like to make something glow about as bright as a 10-watt bulb. That's about 1,000 times brighter, okay? So what we're gonna do is, and you guys get to play along, is we are gonna cut out Lux I through Lux G, and we're gonna put it in PET, where it's gonna be transcribed not with the Lux promoter, with the T7 promoter. That's our project. And it's just one simple operation, okay? So I've written everything down to diagram. If you look at your diagrams in SnapGene, you'll see that PJE202, the arrow face is right. And then in the EGFP map, you'll see the GFP arrow face is counterclockwise, okay? So the one sort of mental trick here is you've got to turn the Lux RNA around, insert it in the right direction so that it behaves the same way as we want, okay? Because there's a ribosome binding site that we're using in front of GFP, okay? So this is the general process. We are going to use, we're going to create just two oligos for this, but each oligo is composed of 2 parts. So in the middle here, I have the Lux PCR product. We're going to cut that out of PJE202. And the product's going to basically be, we're going to have an orange oligo on one end, and on the far end, before the terminator, we're going to have the green one, okay? Those 2 come out of there. And then what we're going to do is we're going to make 2 other oligos. We're going to get the other half of the oligos out of GFP, out of PET, right? And then, oops, we're going to make an oligo product. Shoot. You know what I'm going to do? I'm going to fix this. I did make a mistake. We're going to make this oligo. I have to do this with more colors next time. But basically, we're going to make the orange and the green oligo. Geez, wrong one. We're going to make the orange and green oligo the product of the green part from GFP and the green part from Lux. And we're going to make the orange one also from the other end of GFP and Lux. And so what we're going to have is this long oligo that, we're going to have this long oligo. I don't know how to work this. That's basically from one end of Lux and one end of GFP or a PET, and from the other end of Lux and the other side of PET. Okay. And then we're going to use something called Gibson assembly to put them together, and it's going to work a lot better. So we're going to explain that. Okay. So the steps are, we're going to PCR at the Lux product. We're going to PCR at the GFP, the PET. Okay. When we do that, we're going to get these two middle products. We're going to have a long linear piece from Lux, and we're going to have basically an open circle, which has been cut open. Okay. In practice, and we're also going to, in practice, we did this by taking the same oligo and PCRing out all the way around PET to make this long PCR product that matches perfectly to the Lux product. And then we're going to digest, the plasma with BSE-R1. This is not super important to remember, but if there's any plasma left over from PET after we PCR it, it's just gonna mess up our experiment. So we're gonna use this other restriction enzyme. We're gonna use it to kill and cut open all of the plasma we're not gonna use that we started with. Is that okay? No questions? Okay, and then as described in some coming slides, we're gonna do something called Gibson assembly, and we're gonna pop the PET Lux in. The only trick here is that we actually have to sort of in our mind, just flip over the PCR product so it fits into the PET the way it was, because the plasmids don't care right to left, left to right. They work in either direction, but we have to make sure we're consistent, okay? So that's probably the main thing you might get wrong as we go through. You're gonna actually design the oligos. I'm gonna show you how to design oligos, and then you're gonna design these green and orange oligos. Won't that be fun? Okay, what is Gibson assembly? Okay, Gibson assembly is, to just put it mildly, so much better a way to make a plasmid. It's unbelievable. So here is literally 3 pieces being inserted into a plasmid. The Gibson assembly, this is actually almost so trivial, I'm just gonna miss it. But basically what you can see is, if you can make a PCR product that actually has a little bit of what it's going to join on at the end of it, you can use these identical regions to basically glue the plasma together as you like. Does that make sense? So so I just showed you inserting A into a plasmid. Here I'm showing you a diagram that inserts A and B into a plasmid. Okay. They have done up to 5 or 6 different pieces, all joined in one reaction into one plasmid that transforms into the plate, into the bacteria. That's like fabulous. Okay. So that's just a lot less work, and it works a lot more easily. I'm going to, I think. So I just want to, this is out of order, not in the correct order, but this is how do we join those ends? In the end, what we do is, so you can add so it says add overlap. Sorry, go ahead. Yes? You okay? Oh, someone's just talking to somebody. So what we do is we have this overlap region in PCR, overlap region. And what happens is they use different enzymes. They use an exonuclease to cut back the 5' ends. And this happens on both sides. And then as you basically cool it down, and then the 2 sides join. And then you can actually use a DNA polymerase to fill it in, and then a DNA ligase, and basically it creates one circular plasmid in very high yield. Instead of four bases in a sticky end, it's got like 20 bases in a sticky end. And that actually holds together really well. So that's why the yield of Gibson assembly and CPEC are so much better. The difference between Gibson and CPEC, we're doing CPEC, which is like circular something rather assembly, but the only difference with Gibson and CPEC is in Gibson, you just cut the plasmid open with an enzyme and then you clone directly into it. In CPEC, you actually PCR the plasmid that you want with the proper sticky ends on the end, and then you go from there. That's really the only difference. So they're basically the same technique. I think CPEC works a lot better because you can choose the end of your plasmid. You don't have to design it. Like, it's complicated if you just take some plasmid and you cut it with an enzyme and then you use it for Gibson assembly. You have to work with whatever ends you have, and it doesn't work as well. I think it's not as easy. So we just order it. We can do it at almost the same cost. It just requires one extra reaction. Okay. All right. So I hope everybody, I'm going to walk you through again how to design these oligos, but we're going to talk about, we're getting ready to do that. So I'm going to, these are the 3 rules we're going to remember. You can write them down. But basically, you need a melting temperature of 58 to 65 degrees. Really, 60 to 65 degrees is the melting temperature you want. Both, there are actually 4 melting temperatures in this plasma, in these oligos. There's 2 oligos, each with a half. Each half of the 2 parts of each oligo has to have a melting temperature of 58 to 64 degrees, 65. Okay. Another thing is those temperatures have to match. So we want kind of all 4 temperatures to be about the same. You can make them anywhere in that 58 to 65 degree range, but you want all 4 of the regions to sort of have the same melting temperature. Okay. And then you want to avoid long runs of a single base. So if you got like 12 A's in a row or 7 A's in a row, you might have to move the oligo. And then the last thing is, it's really nice to end the oligo with a G or a C. That helps keep since G's and C's have more hydrogen bonds, they bind nicer. And so they tend to sort of make the end a little neater. Okay. Okay. So now that was actually the results of Paul's work. Is Paul here with us today? Paul did this just the other day. Okay. I'm going to do the PET side. Everybody, I'm going to switch now to SnapGene. Okay, so here's my PET. Okay. Or should I do the other end? All right, I might, depending on the class, I'm gonna do what you want. I'm gonna try just one and see if you want to do the other one, but we'll see. So here's GFP, right? And so it's really nice. You can see the ribosome binding sequences here, ribosome binding sites right here. The MET is the start codon, right? And what I want is I want to detail the oligo here that's going to go to the right. So my mission here, sorry, can everybody—
Ron, real quick, sorry to interrupt, can you show us how you got to that view with the multiple parallel lines of code?
You know what, I'm going to get rid of that. I don't remember how I got this. Oh, you know what? It's this little button with this double-decker button down here, split window, lower left, next to the magnifying glass. I'm going to turn, I find that distracting, but you guys might like it. So looking at this, you can see the EGFP here. That's what I want to replace that. I want to replace that with my genes of interest from PJ2E202. I want to preserve the T7 terminator, right? That's the terminator I have to keep. And I want to preserve the ribosome binding site here. Although it's interesting, I might want to get rid of it. I mean, you can make an argument you want to get rid of it, but we're going to keep it today, okay? So basically, I want to cut out this whole region between the ribosome binding site and T7 and replace it with content from PJE202. And we want to be really sensitive about the spacing. If I add extra bases before or after, that might disturb things, especially on the ribosome binding site side. The ribosome binding site has to be exactly a certain distance from the start codon. Otherwise, it doesn't work very well, right? So Let's start with the terminator. I'm going to just select. See, if I select the terminator, then I click the sequence button below, it takes me right there. And so look, here's the T7 terminator. How nice. Okay. So I want to keep that. And I just kind of want to, I mean, this distance between the stop codon and the terminator, I think that could be useful. So I am going to start with the terminator because I want the terminating, I'm sorry, with the stop codon, the star of the stop codon, I should say stop, but they put a star in there. I want, first of all, which direction do I want the oligo to go here? Left or right? Anybody? Okay, I want to go this way. Okay, because on PET, I want to copy the rest of the plasmid, but not the GFP, right? So I'm going to, in this case, I think I'm going to start with the terminator codon, and I'm going to go to the left. And you can see in the purple there, you should try this. If you do that yourself, you'll see a TM equals in the purple above the cursor. And that's how you can tell how long this thing should be. Okay, I'm going to say, oh, okay, I'm going to go for 64 degrees. Okay, this is a 64, 65 melting temperature. I'm including the stop codon because I believe the stop codon will be the same in both plasmids, in both oligos, and I want kind of PJE202 to stop at the stop codon, and I want the PET one to start at the stop codon. So I'm gonna include it here, and I got a melting temperature of 64, 65. Now I can actually go to the primers. There's a primers menu up and add primer.
I have a question. When you mentioned about the temperature, does it mean that that temperature, like 64 degrees Celsius, will remove this particular section that you highlighted as blue? What's the temperature mean?
The temperature.
TM.
So you remember the PCR part of the lecture? The TM is the annealing temperature for the oligo, right? So that's the temperature at which the oligo will attach to DNA. Okay, and we need that to
detach this particular section that you highlighted.
You have to stay close to your microphone.
That the temperature. Okay, sorry. So the temperature will remove this blue section that you highlighted.
Well, what we're doing is copying the sequence. I wouldn't say it's going to remove it. What we're going to do is we're going to synthesize this sequence, this blue sequence, into an oligomer. And when I have that oligomer, that short piece of DNA, and I add it to PET at 62 degrees, it's going to become fixed in the double helix with this DNA. And then above 62 degrees, it's going to detach.
All right, thanks.
That's what it is. Great question.
Ron, I noticed you ended on a GC on the left-hand side, but not on the right-hand side. Does it matter?
It does matter, but we may come back.
I see. Okay.
What I'm going to do is I'm going to save the spot. I'm going to add primer. In this case, I'm going to the left. So I'm going to take the bottom strand. And you can see that the primer is basically going to the left. So that's a good sign. That's how I know I did it right? It's going in the right direction. It's copying the PET, right? So I'm going to call PET copy term. This is the terminator. Okay. Add primer to template. And now,
did you just annotate it? I missed that. What you were doing? It sounded like you were typing.
Yeah.
Oh, you annotated it. I see. Yeah.
I did. So you can sort of see, I actually have my oligo here on my map, and I can pull back to the sequence and I can find it.
Mm-hmm.
Okay. So why doesn't everybody try to make a PET copy terminator? And you can choose your own temperature. We may go back and change it later. Okay.
When I add primer, it asks me whether I choose the top or the bottom strand.
That's right. So if you choose the top strand, it's going to go clockwise. you choose the bottom strand, it's going to go counterclockwise. So I'm going to ask you to figure out which of those two you need. But
Pardon me, I got a question.
remember, these oligos are going to be going here all the way around to here. So this one's going to be the top one, and it's going to go clockwise all the way back around to here. Okay, so the oligo on the initiator side and the star codon side is going to go around away from GFP, and the one on the left hand side near the terminator is going to go counterclockwise away from GFP, right? So that should help you figure out what's its top and bottom. No? It's not a test, but I want you to spend a minute looking at it and figuring that out. When you do the oligo, when you do the primer, oops, you'll see that actually the primer direction from top or bottom is actually in the diagram, okay? So you can test it. Try it out yourself, all right? I think one other thing I didn't tell you is I like shorter oligos. The reason is they're cheaper. But, you know, if it's going to work better at length, you might want to do longer. I mean, you're not going to buy these, so it's probably okay. Choose your own temperature.
Wait, can I ask something?
Sure.
Oh, okay. So I can't tell if anybody can hear me because
I can hear you.
oh, so I didn't. I think I may have missed something. Is the temperature that it anneals is affected by what? The length of it or something?
Mm-hmm. And the sequence. Like sometimes to get 60 degrees, it's got to be pretty long. Sometimes it's pretty short. It just depends on the actual sequence of the DNA that you're copying. It's kind of weird.
So this thing will tell you.
Uh-huh.
This thing will just tell you if you highlight it what the temperature is going to be.
That's exactly right.
Is that?
Yeah.
Okay.
Here, I'll do this. I'll do it again right here on the origin. Like I'll just like start copying here. Like after about like 10 bases, it starts giving me a temperature. In this case, for the F1 origin to copy it, to get to 60, I got to go all the way out here because there's a lot of A's and T's there. Yeah, that's not really great, but so it is. How's everybody doing? Oh, sorry.
So once we highlight it, then what do we do?
There's a, you can't see it on my window, but on the menus at the top, there's a menu called primer, and you can just select add primer. And then we'll ask you whether you want the top or bottom strand. And you got to name it something. I named mine pet copy term, but you can name it what you think helps you understand.
And top strand goes which direction?
Clockwise. DNA is kind of made to be read. So we read it left to right, just like we read regular writing. And it turns out that the RNA polymerase also copies in that direction. Everybody got it? A few more minutes?
Somehow it did not give me the chance to name it though. It just take it.
I'm gonna do it again. Okay. I'm just gonna do this. I'll go to 65 degrees this time. Okay. So then I've selected this, it's blue, and then I go up to the primers menu, which you can't see, and then I select add primer. And here it is. It says, do you want the top strand or the bottom strand? I select bottom strand. And then you can see in the picture the primer goes to the left, away from the GFP gene. Okay. And then I can name it up here where it says primer 1. And if you want, you can type a note in there to description. And then on the lower right hand corner, it says add primer to template, and then it's included in your file. Actually, you can see on this bar at the bottom, there's actually a list of your primers. There's quite a few primers that came with the PET itself. Everybody feeling good? This is the tough one. Your first primer is always the hardest. There's my PET copy term. There's the melting temperature here, 62 degrees, the date, the number, the sequence, and the fact is 25 bases long. Okay. It looks like some people are still working. How long do we have, Doug? It's 4 o'clock now.
Yeah, we probably want to maybe stop at 4:30, I would think.
Okay, guys, I've shown you how to do this. I'm going to walk through the first oligo, okay? And then I'm going to leave it for you guys to do after, because I still have more talk to do. Does that sound right? Or do you guys want to really go through this carefully? I'll let the class decide who wants to. I don't know how to vote, but who wants to just do the oligos and who wants to listen to the rest of the talk about other synthetic biology?
The rest of the talk.
Okay,
Yeah,
no one else is voting. Okay. All right, I'm going to do a quick demo on this. Okay, so all right, so I'm going to go to the other window. Okay, I'm going to go to the window. Where's the, okay, share. Okay. So in this case, what I want is something that is by the terminator. So we can see here that I want to go from Lux I to Lux G. And so this is the terminator here. So I'm going to pull up the sequence here. And I'm going to take the terminator again. And now I'm going to go into the gene, right? So P-Lux is going to, this oligo is going to end at the end of Lux G. And then I'm going to attach it to the oligo for the end of PET, right? So let's see, I think I went to 62 degrees here. Wow, so I have to go a long way. So there's 64 degrees. I think it's the temperature I took. And I'm going to do a primer for that. And in this case, I still want to, in this case, I'm going to take the top strand. Why am I taking the top strand? Well, I'm going to attach that to the same oligo I just made where I took the bottom strand. Why did I have to do that? Does anybody know? It's because the arrow is pointing the other direction. Remember, the gene can be one direction or the other on the plasmid. And these two plasmids they're pointing in different directions. So I kind of got to remember to turn the plasmid, turn the oligo around. Okay, so now I've done that. Right, so there we go. There's my, that's the top strand. Yeah, that's the top strand. All right, so then what I'm going to do,
do you choose the two direction because you want to put two genes so that they can go left and right from the same spot?
I'm gonna go back to my deck here. So these oligos are a little bit strange. They are each composed of two halves. One of them from PJE202 and the other half from the PET vector. So I'm going to take those two sequences. I'm going to put them together here.
Okay.
Yeah? Okay. So in fact, maybe I'll just do that right here. So I'm going to let's go to the primer. You know, and so I'll just copy them. So this is PLux on the terminator. And here's the PET primer, PET copy term. And I'm just going to copy that. Okay, so this is the one that's purple and green. And so one thing you can see is that they both have TAA at the beginning and end. I don't have to have both copies of that. So because if I put these together like that, then this side will use all of these sequences and they'll both use 3 of the bases. Okay. So that is oligo number 1. Does that make sense? It's actually using the oligos to actually attach the two plasmids instead of overhanging bases. That's what the genius of Gibson is. Okay. It may not be completely clear. These things are usually not perfectly clear the first time through, but you can just continue the exercise. Maybe I can come back and talk about it later, or we can do Q&A, or Doug can answer questions. Okay.
You have one more question. It's kind of curious. How do you able to choose these two particular sections to join together? But this might be.
Oh no, there's actually a company that's going to make this. They actually have a machine that will make this oligo this long, and I just put it into a website and I order it and they send it over to the lab in like 2 days.
Okay.
Isn't that great? Right. So this is actually an experiment. Paul Ferguson did this. He used 2 different polymerases. We made Fusion ourselves here at Laney, which is a pretty good polymerase. And then there's Q5 polymerase, which is a really long. It specializes in very long sequences. And so the first lane and the last lane are size standards. That's a 1 kilobase ladder. The bottom one is 1,000 bases, and then every line up is another 1,000 bases up to where I think 10 or 12,000 bases. And then on the right-hand side are 100 base ladders, where I think the top one is another 1,000 bases, and every ladder down under that is 100 bases less. Okay, so it tells you how long things are basically. The first 3 reactions are done with Fusion, and all we see are the primers. Those are the DNA, the oligos that we ordered, and nothing's happening there. And it looks like Fusion, the Lux I through G segment is about 3,000 bases long, and so is the plasmids about 4,000 bases long. So it's probable that the polymerase just simply can't run that long, and so we were planning to use Fusion, but looks like we can't. And the Q5 polymerase, 2 of the oligos are successful. So the PET oligo is pretty successful. It just picks the PET right out. The PLux oligo in the lane, what is that? That's lane 7. That didn't work, but for some reason the PET Lux combo. So I designed 3 oligo sets. One of them just for PET, one of them just for PLux or PJE202, and one set to make the CPECs that's combined. And that one works, even though it looks really messy We got to work on cleaning it up a little bit, but we did get a fragment of the right size at the top there. So we're ready to go. And hopefully next time I give this talk, we'll actually be able to show the bacterium glowing. Okay. So that's great. That's Laney work we're presenting. Good job, Paul. Okay. So I'm gonna just jump. So once, and just to sort of fill in the details, once we actually have the plasmid, we transform bacterium that will basically copy the plasmid, and then we can use the plasmid to purify more of the plasmid. And so this is a fairly simple workup called a mini prep. You can do in a couple hours with a centrifuge and a little bit of cell culture, a little bit of E. coli, and you can get a fairly large amount of plasmid DNA out of there. And then, just like I showed you, we check everything with an agarose gel to see. Okay. Okay, so that's sort of like that process. What I'm now going to talk about some other extensions to biosynthetic pathways. So that was a very simple pathway, which had 3 enzymes in it that produce light. This is a violicin pathway. This is this is artemisinin. There's a group here at Berkeley that's very famous, and they produced this stuff called artemisinin. Artemisinin is actually a great antimalarial compound that's really it's produced in I think a tree bark or something like this. It's really hard to get any amount of compound out, but it's been proven to be effective against malaria, which is more and more resistant to the common treatments. And they produce this stuff in yeast for just a few pennies a dose. Each one of these arrows represent a step, that biosynthetic step that was produced from an enzyme. So they actually put in every enzyme that did all these chemical transformations to the final product. They did this in yeast, and it took them several years to do it. And it was a whole team of 20 people. And it was a massive project. They started a pharmaceutical company from this, based on this. But that's how large the projects can get. And it's becoming, there are now teams that are doing things like morphine and other very complicated compounds. We can produce our pharmaceuticals from yeast and don't have to like extract them from roots and things like that we find that. All right. This is the violicin pathway. This is a pathway of just 5 genes. This stuff reportedly costs like $100,000 a pound. And so the idea that you could just manipulate the pathway, put that into yeast or E. coli, and produce this extremely valuable anti-cancer drug is just fascinating, right? That just shows the breadth of the kind of work that you can do. There's actually a company that released the violicin genes and let people experiment with them. And then you can actually see in each different step the compound that was made and the color that it had, because violicin is deep purple. Okay, there are large numbers of, there are places where you can actually look at all the genes humanity has ever sequenced. There are millions of them. And so basically the opportunities here are unlimited. Everything that the drug companies have, pretty much you can have too. The question is, what do you do with it? This is some bioluminescent projects that inspired us. This team from Cambridge, they have these glowing fluorescent flasks. Now, after we played with PJE 202 a little bit, I understand that to take these pictures, they probably had to open the shutter for 30 seconds. But they were able to modify the luciferase to produce different colors, which is fascinating. So that's protein engineering, protein design. There are many different ways that we can take the system once it's working and tweak it to try to improve better and different light. The right-hand picture is a picture from the Glowing Plant Project, which is now dead, but the idea was to make plants that glow so brightly at night they can replace street lights. A fascinating topic that I'm sure the project will come back. So this is actually from this project. Okay. In my history, I helped the open source insulin project go by releasing a copy of insulin through one of the startups that we had. That's the first result coming off the assembly line. That's the original clone from a DIY bio open source insulin, right? And then there are many, many other things that have been doing in academia that are very impressive. This is an imaging system where people can shine light on a plate and make photographs with photographs of using different frequencies of light. This is a computer that was built that will take sequences of light and use it to program the production of genes. And all kinds of oscillators and all kinds of crazy devices are being built to try to emulate electronic signaling in terms of gene expression. So that's what's going on in the academic world. Those slides are somewhat old, and they're quite. There's something new happening all the time. Just have 8 more minutes. Doug requested that I share this. This is a blog post that just came out very recently, like Friday, I think. This is a plot of lab-grown meat, so cell culture technology. These companies are doing extremely well. Some of you may have heard of Moore's Law. Moore's Law is the blue line at the top. And as you can see, the cost of a transistor every year drops. Every 2 years or so, it drops in half, maybe 3. And that's what has created the entire internet and all of IT and all these giant internet companies. That's the pricing law that has technology that's driven everything almost everything in tech that we see. This is the red line. The green line is actually the price of a human genome sequence, which is dropping tremendously, and it's affecting our lives all the time, but it's not really a consumer product. There's no internet of gene sequences. It definitely affects synthetic biology though. The red line is a bunch of dots where the price of a serving of lab-grown meat was promised, was announced. And I saved those and plotted them on the same plot. And you can see that the biotechnology to produce meat from a bioreactor is dropping many times faster than Moore's Law. In fact, it's only been 7 years, and some people are pronouncing the price to be incredibly low, like getting closer to $10 a serving now. And so even though it feels like a pie in the sky kind of project, it's likely that we'll actually have consumer goods on the market in the next 2 or 3 years from now. So that's hopefully you've enjoyed that. So that's my talk.
Well, Ron, thank you so much. You've given us a lot to chew on, and I know I've got my homework ahead of me. I'm going to be pouring through those maps that you've helped us start and getting more familiar with how to do CPEC, Gibson cloning.
We're going to be doing our first CPEC hopefully next week. And
that's right.
That will be a lot of fun. And then if things go well and that we get the system working, I'm not sure it's going to work this round, but it may be next round, we'll get it to work, and we will, we'll be able to modify the system to try to improve its output this fall, we hope.
Exactly. Yeah. And this is going to be a core part of our curriculum for upcoming certificates. So what you've laid out here will be a big module. And what we're going to cover in fall 2021 is how to do this type of cloning and molecular bio. So thank you again. Any other questions out there from anyone who's been watching and listening? Any questions?
Are there SnapGene tutorials online, like some kind of classes or videos or stuff for how to use the program more?
There definitely are. I've seen a number of them on YouTube. If you check it out, there's a channel on YouTube for SnapGene.
Yeah, we may have to add just like an hour of working with SnapGene separately from this talk. It's just.
I think it'd be a great idea. Yeah.
We might come back later and do that if that's what you guys want to.
I think that's a great idea. And then I have one more question. If we have the associates in biomanufacturing and we want to try and work somewhere where we're working with SnapGene or with transformations and plasmids, and who do you think's hiring around here, and what roles should we go for?
There are a lot of tech roles available in startups. I mean, one of the great things about Laney is Doug has been placing students in these startups. And I know quite a few of them. I think the important thing is to just get your basic techniques down. All these companies want to see someone who's done these things with their hands. And that's one of the great things about taking the class here. Generally, Indeed and Craigslist have a lot of ads for these kind of things. Some of the companies that are affiliated with Laney will send Doug an ad though.
Yeah, absolutely. So definitely, Brandon, you can follow up with me. I know we're already connected, but anyone else who's listening too, feel free to reach out. I've got LinkedIn, connect with me, definitely, because both myself and Ron, we have pretty big networks in the Bay Area. So if you're looking to connect to people who are in the industry, use LinkedIn. That is the way.
LinkedIn is a great way to see what's going on. A lot of people post, I'm hiring, up there in their profile. Okay, all quiet. All right, guys. It was a pleasure.
Yeah, Ron, thank you so much again. And I'd love to follow up with you at some point. We could probably schedule a time to do another kind of SnapGene intensive or something.
Yeah, it looks like I need to make material just about using SnapGene. Yeah, I'd love to do that.
And if you don't mind, I might, if you don't mind sharing the maps that you've developed, maybe we could talk about this later, but if you have any kind of preliminary or like these maps you were showing us today that you had the different oligos set up for.
Oh, those are the same. Yeah, let's validate them in the lab.
Okay, right. Yeah, let's not give out bad information. Yeah.
I mean, I sent you guys. I'll be happy to send those out.
Yeah. Okay, great.
But I would also like you guys. It's the only exercise I have.
Yes. Yeah. No, it's true. Yeah. We don't want to ruin the exercise.
We'll send the oligos out.
Yeah.
Okay.
Cool.
All right.
Great. Thank you so much again.
Brandon. Thanks, Emily.